Hi, my name is Katie Lample, and I am a Master's of Science student in Fisheries Sciences in the Warnell School of Forestry and Natural Resources. And today I'm presenting to you preliminary results of my master's thesis project with co-authors Dr. Susan Wild and Dr. Lori Sutter. And this is a coastal incentive grant from the Georgia DNR Coastal Resource Division. And we are evaluating the application of a Regional Hydrogeomorphic Approach Guidebook to Georgia salt marshes. And so the Hydrogeomorphic Approach, or HGM, is intended to be a rapid assessment that the U.S. Army Corps of Engineers uses for regulatory purposes. And it has been a useful tool for evaluating geomorphological processes as well as for habitat functions. It's been used for wetland replacement, and it has been a successful approach in restoring hydrological functions. However, the U.S. Army Corps of Engineers Savannah District recently required the use of this regional guidebook for applying the approach of tidal fringe wetlands along the Mississippi and Alabama Gulf Coast to assess in Georgia salt marshes. And this particular guidebook is for microtidal marshes, which are less than 2 meters in tidal range, and we want to test the applicability to macrotidal systems that are found in Georgia salt marshes, or salt marshes that are more than 4 meters in tidal range. And so in particular, we are testing this assessment on Little St. Simons Island, Georgia. And Little St. Simons Island is a privately owned island and one of the least developed of the barrier islands of Georgia. It's known for its diverse wildlife and habitat, and there is a lot of ongoing research of rare and threatened species, including many shorebirds, bats, and green sea turtles. And so to give you a little bit of history of Little St. Simons Island, in the 1930s, the Civilian Conservation Corps created these parallel grid ditches in an area of the island. And they were used for managing mosquito populations as well as for rice cultivation. And these channelized areas are still present in the salt marsh today, and they have resulted in impaired hydrological altered connectivity within the salt marsh habitats. And preliminary analysis of water quality, vegetation, and fish have indicated reduced ecosystem function in these channelized areas. And so our goals is we want to evaluate a potential ecological lift or difference in degradation between this hydrologically altered area of Little St. Simons Island and a reference or unaltered area of the island. And so we want to determine if these areas or creek sheds are sinks or sources of sediments and nutrients. We also want to develop seasonal tidal budgets in each of these creek sheds, and then we want to evaluate the HGM approach for coastal Georgia as a rapid approach that can accurately predict relative ecosystem function. And so our objectives were we wanted to compare a seasonal field study to the HGM approach. And so essentially comparing two forms of assessment, the HGM to a more field case ground truthing study. And our hypotheses were that there would be significant differences between this hydrologically altered creek shed versus the unaltered creek shed. And for today, I'm going to focus on the biogeochemical cycling function of the HGM, comparing to our field case study focusing on nutrients, sediment, and plankton dynamics, as there are multiple functions to the HGM approach. And so our other objective today was we wanted to assess the seasonal plankton community structure with incoming and outgoing tides. And with that, our hypotheses were that a freshwater discharge will enhance nutrient loading and algal productivity, as well as may result in harmful algal blooms being more prevalent in these hydrologically altered marsh areas. And so our study area, as I mentioned, there are two creek sheds we have on the island, and we have an altered area of the island and an unaltered area or reference site of the island that's untouched. And so we have two study sites, and we are doing seasonal sampling for this project. We have already done fall and winter sampling campaigns. And so here's a more zoomed in area of the island, and our study areas are outlined in lime green, and you can see the ditches within this altered area outlined in red. And this is a more zoomed in area of our altered site point, and then also our reference site. And right above this altered site is the Altamaha area, Altamaha River, or also the freshwater source to the study areas. And so for our methods, we wanted to collect discharge, and by doing so, using an acoustic Doppler current profiler, or also known as an ADCP, which I am very fortunate to be working with Dr. Jay Shelton and Wesley Guerin on this project, and they have allowed us to use what's called the HyCat, which is an autonomous surface vehicle. But for this project, we are using it to traverse the creek every 15 minutes for a full tidal cycle for 4 transects. And we also collected water samples every 30 minutes for suspended sediment concentration, dissolved organic carbon, nutrients, and phytoplankton as well. And we also filtered for nutrients in the field before putting them on ice. For water quality, we deployed an EXO at the site that collected data every minute, and it collected for all water quality parameters, as well as it has a special total algae dual sensor on it that measures chlorophyll-a and phycocyanin, so we can measure the amount of algal biomass in the water, or chlorophyll-a, and then determine how much of that algal biomass is blue-green algae, or possibly cyanobacteria, by measuring phycocyanin. And so to show you some preliminary results, I have 2 figures here, and on the left-hand side, I have a figure for the altered site, and on the right-hand side, I have a figure for the reference site. And all figures that I'm going to show you today, I will have time on the x-axis for the full tidal cycle that we collected from low tide to low tide, and then on the y-axis, I will show different inlets that we are comparing today. And on this figure, you have green points that are indicating when the tide is incoming, and you have blue points that are indicating when the tide is outgoing, and black is indicating suspended sediment concentration. This analysis is only for our October data. We are still working on our December data, so just focusing on October data today. But here I have suspended sediment concentration comparing to average Qs. So when we took discharge over the 4 transects every 15 minutes, I averaged the 4 discharge measurements over the full tidal cycle. So here we can see that when sediment is coming in the incoming tides, and then how it kind of decreases as the tide is outgoing. And here on the right-hand side with the reference site, where again I have suspended sediment concentration on the secondary axes, and how we're seeing not so much sediment coming in with incoming tides compared to our altered site. And so similar figures, just with different y-axes and secondary axes. Again, we have chlorophyll coming in on this figure, and black is ammonium, and again this is for October sampling. And so we have our altered site on the left-hand side and our reference site on the right-hand side over the full tidal cycle, and we can see that more ammonium is coming into the creek sheds, and also more is going out of the creek sheds compared to our altered sites, as well as we have more chlorophyll or algal biomass coming in as well as outgoing compared to our altered sites. And these figures represent chlorophyll once again. And then on our secondary y-axis this time, we have nitrite or nitrate. And again, we have time on the x-axis. And here we have, again, the altered site and reference site comparing. And on the altered site, we have some nitrite or nitrate coming in following a similar trend with the chlorophyll or algal biomass. And then on the reference site, we have the chlorophyll again increasing more as it's incoming compared to our altered site and more outgoing. And for our nitrite data, we're still in preliminary method development, so we are missing a few data points for our nitrite data. However, I wanted to still kind of demonstrate what we're likely seeing in the reference site and how it may be decreasing more in the reference site compared to the altered site. And then finally, we wanted to compare our EXO data to our chlorophyll data, where we also extracted for algal biomass in the lab in micrograms per liter. And so we regressed the EXO data. So we wanted to build confidence in our EXO data, which again were collected every minute. And here I have put the EXO data at the same sampling time point as when we collected that water sample with the chlorophyll. And so they're at the same time point, so we're able to compare and see the relationship between these two sampling techniques. And as you can see there, we have an acceptable R² for both of the sites, and how now, since our relationship is rather strong, we can trust the data from these sensors. So in summary, we are seeing differences between each creek shed, and I have a table for you here. And so we were seeing in the altered site where more was coming in for sediment and compared to our reference site, where so sediment may be a source, a sink in our reference site, and then possibly a source in our reference site, as well as we saw for algae or for algal biomass, we saw more coming into our reference site and more going out compared to our altered site as well, indicating this may be, algae may be more of a source, so more is going out for outgoing tides, possibly compared to our altered sites. And for ammonium, we had more coming in and more going out at our reference site. So maybe ammonium, I have here maybe sink or source in October as well compared to our altered site. And then our nitrate may be more of a sink or more storing in our altered sites compared to our reference site. However, we did not have enough data to demonstrate that, so it's unknown right now to kind of hypothesize whether or not that nitrate may be a sink or source at our reference site. And so we were also able to see that ammonium may be more available than nitrite and nitrate, and following similar trends with algae. Also, algae in our reference site, there was less nitrite, possibly less nitrite and less nitrate at our reference site, and therefore algae or may be consuming more of this available dissolved nitrogen at the site compared to our altered site. And so we were also able to validate our chlorophyll-a and EXO measurements and have confidence that that we can trust our EXO data compared to lab extraction concentrations. So what's next? Well, we hopefully will get our hands on a flow cam to rapidly analyze algal taxonomic composition, species abundance, as well as biomass, as I did not have that data today. And then we want to compare algal biomass to further nutrient analysis over the seasons, different seasons. And we then want to calculate tidal budgets for inputs and outputs throughout the full tidal cycle in order so we can calculate that budget. So then we can compare our altered sites and unaltered sites and determine a ecological lift between the sites. And then we want to then compare that to our HGM functions, as there are several functions for this assessment. So that is all I have for you today, and these are my acknowledgements, and I want to thank Dr. Brian Bledsoe and Dr. Brian Irwin, who are my committee members, as well as Dr. Jay Shelton and Wesley Guerin and several others for contributing to this project so far. And if you have any questions, my email is listed here, and thank you.